Hello.
Yes, ma'am.
Good evening.
Good evening, ma'am.
Good evening. Only six students are there. They know it's 6 p.m., right? Should we start now? Hello. Uh, I think we should start actually. Should we start?
Yes, ma'am.
Yeah, but only 7 people are there. They all know it's 6 p.m., right?
Yes,
Timing.
ma'am. Yes, ma'am.
Okay, but still 7 people are there. Okay, we'll start because there are 4 units. It's so difficult to finish actually within this small period of time. Actually, we'll study there are four cells and biomolecules. There are 4 units actually. Unit 1, it contains cell and cell organelles. Unit 2, carbohydrates and lipids. Unit 3, proteins and nucleic acids. Unit 4, intermediary hmm, metabolism. So we'll start the unit 1, cell and cell organelles. Am I audible?
Yes, ma'am.
Okay. Cells and biomolecules, and in this topic we will learn about the it describe the
ମ୍ୟାମ୍
structure of
ଭିଡିଓରେ କିଛି ଆସିବନି?
ଭିଡିଓରେ ତ ପିପିଟି କିଛି ବନା ହେଇନି କିଛି
ଓହୋ।
ବୁଝିଲେ।
ନାଇଁ ଆଗରୁ ଯୋଉ କ୍ଲାସ ଗୁରା କରୁଥିଲୁ ପିପିଟି ବନାଇକି ସ୍କ୍ରିନ୍ ସେୟାର ହେଇକି ଆମେ ପଢୁଥିଲୁ ତ ସେଥିପାଇଁ ପଚାରିଲି।
ଆଚ୍ଛା ଏଗୁଡାକ ତ ଏତେ ସର୍ଟ ଟାଇମରେ ପିପିଟି ତ ମୁଁ ବନେଇ ନେଇ କିଛି
ଆଜ୍ଞା।
ପିପିଟି ହେଇଥିଲେ ଆକ୍ଚୁଆଲି ଟିକେ କେମିଷ୍ଟ୍ରି ଅଛି okay, cells and cell organelles. As you know, the cell is the basic unit uh, uh, of the life present on our earth. We'll, after studying this unit, we'll actually learn the existence of the cell as the basic structural unit of living organisms, the main domain of life, distinguish between major types of cells, differentiate between the plant and animal cell. So first we'll know the biochemical composition of the cell. What is the cell composition? Out of nearly 50 or so elements in the most living organisms, 6 elements, namely oxygen, carbon, hydrogen, nitrogen, and phosphorus, constitute 99% by weight of a living organisms. Chemical composition of the cell: oxygen 68% by weight, carbon 18%, hydrogen 10% in the cell, nitrogen 3%, and elements present in the cell are sulfur, potassium, sodium, chlorine, magnesium. Water constitutes in the cell nearly 60 to 80% of the weight of the living organisms and is rightly called the solvent of life. In the earlier, actually, do you have the material with you? Hello? Hello?
ମ୍ୟାମ୍ କଣ କହିଲେ?
ତମର ମେଟେରିଆଲ ତମ ପାଖରେ ଅଛି?
ମେଟେରିଆଲ ମାନେ ବହି ଅଛି ଆଜ୍ଞା, ବହି ଆସିଥିଲା ପୋଷ୍ଟାଲରେ।
ଆଚ୍ଛା ଆଚ୍ଛା, ସେ ବହିଟା ଅଛି ପାଖରେ?
ଆଜ୍ଞା ଆଜ୍ଞା, ବହିଟା ଅଛି ଆଜ୍ଞା।
ଆଚ୍ଛା ଆଚ୍ଛା, ଆଉ ମୁଁ ଯୋଉ ଗୁଡ଼ା ଷ୍ଟ୍ରକ୍ଚର ଗୁଡ଼ାକ ଯୋଉ ପଡ଼ିବ, ମୁଁ କହିବି ସେଇ ପେଜଟା ଟିକେ ଦେଖିବ ଷ୍ଟ୍ରକ୍ଚର ଗୁଡ଼ାକ,
ଆଜ୍ଞା
କେମିକାଲ
ଆଜ୍ଞା
ଷ୍ଟ୍ରକ୍ଚର। ହଁ ହଁ ହଁ। What is the domains of life actually? Earlier biologists believed that all the life of earth belonged to one of the two primary lineages of eukaryotes. What is eukaryotes? Eukaryotes, which include animals, plants, fungi, some single cell organism, and prokaryotes, which what is prokaryotes, which include bacteria and all remaining microscopic organisms. So according to the Carl Weiss system of classification, tree of life consists of three domains: archaea, bacteria, and eukarya. The archaea and the bacteria are prokaryotic single-celled organisms whose cells have no nucleus. Cells of eukarya possess nucleus and membrane-bound organelles. The plant, animal, fungi, protozoa are included in the eukarya. Structure of the prokaryotic cell. Prokaryotic and eukaryotic. Prokaryotic cell are the single cell microorganisms known to be the earliest living organism on the earth. Prokaryotes include bacteria and archaea. The characteristics of the prokaryotic cells are that they lack a nuclear membrane and membrane bound organelles such as mitochondria, Golgi bodies, chloroplast, lysosomes, etc. The DNA present in the region in the cytoplasm without membrane and is known as the nucleoid. It contains actually prokaryotic cell contains cell wall, nucleode, plasmids, ribosome, flagella. Flagella is the structures are composed of protein giving cells capacity to move. Pilus, capsule. These are the structure of the prokaryotic cell. Structure of the eukaryotic cell. Eukaryotes have clearly defined nucleus. They have the nucleus. There contains organelles including mitochondria, endoplasmic reticulum, a Golgi apparatus, and lysosomes. Prokaryote and eukaryote. There is difference between the prokaryotes and eukaryotes. Prokaryotes contains bacteria, blue-green algae. Eukaryotes contains protozoa, algae, plants, and animals. Prokaryote cell size 1 to 10 micro in diameter. Eukaryotes 10 to 100 micrometer in diameter. It has nucleus present absent. Prokaryote nucleus is absent. Eukaryotes nucleus is present. Cell division occurs in the prokaryotes in the binary fission. It is not in the eukaryotes. Chloroplast absent in the prokaryotes. Chloroplast is present in the eukaryotes. So these are the difference between the prokaryotes and eukaryotes. What is cell membrane? Cell, whether prokaryotic or eukaryotic, are bound by limiting membrane called plasma membrane. In the plant, cell membrane covered by a rigid cell wall in which the major component is the cellulose. In fungi, the cell wall is made of the chitin. What is nucleus? All the eukaryotes, the nucleus represent most prominent and conspicuous organelle of the cell. The nucleus being the control center of the nucleus is the control center of the cell, composed of DNA associated with a set of 5 basic proteins called histones. Nucleus, which contains 5 basic protein, it is called histone. The nucleus filled up with the fluid, it is called nucleoplasm. which contains a rough spherical body nucleus within which the RNA component of ribosomes is synthesized. And endoplasmic reticulum. What is endoplasmic reticulum? The cytoplasm of most eukaryotic cells contain a very complex network is called endoplasmic reticulum. There are two types of endoplasmic reticulum. One is rough endoplasmic reticulum, another is smooth endoplasmic reticulum. Endoplasmic, these are actively involved in synthesis of proteins that are meant to be exported out of the cell and become part of the membrane. Eukaryotics, by contrast, is not associated with the ribosomes and its functions is different from the endoplasmic reticulum. What is Golgi bodies? Golgi bodies appear within the cell as starch and flattened vesicles. Carbohydrate residue on the protein in a process is called glycosylation. Such glycosylated proteins are recognized at different sites within the cells. Golgi bodies thus play a key role in chemically modifying newly synthesized proteins. Proteins secreted from the Golgi apparatus are directed to lysosomes or to the cell membrane, still others are designed for secretion of the cell exterior. Then mitochondria, what is mitochondria? Eukaryotic cell of living organism continuously carry out a huge number of chemical reactions to live, grow, reproduce and fight off diseases. All these processes require energy at the cellular level. Mitochondria are the largest organelles in a cell with a diameter of the 0.5 to 1 micrometer and their number vary from one cell type to another. Mitochondria are of different shapes, spherical in the liver cell, cylindrical in the kidney cell, or thread-like in the fibroblast. Chloroplast, one of the most distinguishing features of all eukaryotic plant cells is that in addition to mitochondria, they contain special light harvesting organelles known as the chloroplast. These are the largest organelles in the plant cell. Chloroplasts are the largest organelles in a plant cell, are about 5 to 8 micrometer long and 2 to 3 micrometer thick. About 50% of the dry weight of the chloroplast are protein. And the rest is mostly lipids. The lipid material of the chloroplast contains chlorophyll, carotenoid, phospholipids. Chloroplast, like mitochondria, possess bilayer membrane. The difference between the animal and plant cell. Animal cell wall is absent. In the plant cell wall is present. It called cellulose. Animal cell membrane present. Plant cell, it is also present. In both plant animal, cell membrane present. Animal cell flagella may be present. Plant cell absent. So these are the difference in the page number 24. It's the difference between the animal and plant cells that is there. So in this unit, we know about the plant animal cell differ in some respect of each other, that is exterior structure, interior structure and cell organelles. And various organelles of a cell can be fractionated by differential centrifugation method, which based on the difference in the density of the organelle. So, and in the unit 2, it is carbohydrates and lipids. Carbohydrates form the most abundant class of the organic compounds. They are compounds containing carbohydrates, both contain carbon and hydrogen. They are the compounds containing carbon, hydrogen and also oxygen. Carbohydrate, it contains oxygen also. Hydrogen, oxygen in these compounds are generally present in the ratio 2 to 1 atom. There are number of monosaccharides, disaccharides, oligosaccharides, polysaccharides are there. There are different types of carbohydrates. Carbohydrates can be divided into classes. according to their size of the molecule. Monosaccharide, basic unit of carbohydrate, simple sugar, CnH2nON. Disaccharides consist of two monosaccharides. When two molecules of monosaccharides are joined, it gives disaccharides. And oligosaccharides are number of include the sugar with 10 monosaccharides. When 10 monosaccharides attach each other, it is called oligosaccharides. Polysaccharides are made up of monosaccharides joined together but contains thousands unit. Polysaccharides are thousands of the monosaccharide, but oligosaccharides are 10 number of the monosaccharides. Protein are the complex, diverse and high molecular weight polymers found in the cell. Monomeric unit of proteins are called the amino acids. Monosaccharides. Carbohydrates, it is divided into three groups: monosaccharides, disaccharides, and polysaccharides. You can see the page number 38. You can see page number 38, monosaccharides. Can you see the page number 38? 38 page number. Carbohydrates are divided into monosaccharides, disaccharides, and polysaccharides. Monosaccharides contains aldose, glucose, ribose, galactose, and ketose, fructose. Disaccharides, sucrose, maltose, lactose. Polysaccharides, starch, glycogen, cellulose. The monosaccharides. And what is glycosides? Glycosides are when, since the monosaccharides are aldose and ketose, which contains alcohol, monosaccharides it contains both aldehyde group and keto group. Within the same molecule, they have alcoholic group and aldehyde group or keto group. The two group combine intramolecularly. The aldehyde group and the alcohol group combined intramolecularly to give a cyclic hemiacetal. Give the six-membered cyclic hemiacetal or aldoses and ketoses are quite stable. For example, D-glucose, the hemiacetal will be formed between the aldehyde group of carbon 1 and alcoholic group of the carbon 5. The carbon 1 and carbon 5 they join, it gives the alpha form and the beta form. So it is in the page number 40. You can see the reaction. Open chain aldehyde form of glucose, the carbon 1 and carbon 1 attached to the OH group, keto group, and carbon 5 contains OH group is attached there. Then they cyclize to give the cyclic hemiacetal to alpha form and beta form. The cyclic hemiacetal formed by a monosaccharide can react with the alcohol to form the acetal or ketal. When the D-glucose treated with the alcohol, ethyl alcohol, it forms ethyl alpha D glucosides and ethyl beta D glucosides. You can see the page number 40. Then amino sugar. D-galactose as well as the D-glucose exist in the nature as their two amino derivatives are commonly referred to as the amino sugar. Amino sugar are formed when the hydroxyl group of the OH group, the OH group of the hydroxyl group replaced by the amino group NH2, it forms the amino sugars. Glucose form beta D glucosamine, and by a similar reaction galactose also form beta D galactosamine. When the OH group of the glucose and galactose treated with the amine, it forms the alpha D glucosamine and beta D glucosamine. Similarly, beta D galactosamine is an important component of the carbohydrates. And deoxy sugar. When one or more hydroxy group of the monosaccharide replaced by the hydrogen, as you know, the carbohydrates contains both the hydrogen and the hydroxyl group. When the hydroxyl group OH groups, the OH group replaced by the hydrogen, it is called the deoxy sugar. The deoxyribose is one such derivative in which hydroxy, the C2 is replaced by the hydrogen. Example, fructose is deoxyderivative of the glucose, in which CH2OH become CH3. It is secreted in the mother's milk, helps in the development of the immune system of the newborn babies. Fucose, FUCOSE, fucose. It is common constituent of the mammals, insect and plant cell surface. Rhamnose is the deoxyderivative of the mannose, which occurs naturally as L-sugar. It is more commonly found in plants and bacteria than animals. Structure, it is given in the structure, page number 41. L-fucose, L-rhamnose, 2-deoxyribose. Then disaccharides. As you know, the two monosaccharides, when combined, it gives a disaccharides. Disaccharides are most commonly occurring oligosaccharides in nature. They yield two monosaccharide molecules on hydrolysis. A disaccharide is formed when monosaccharide get connected by glycosidic glycosidic bond formed between when two alpha D glucopyranose and alpha D glucopyranose. Two monosaccharides, when they got hydrolysis, one oxygen, they joined together with a oxygen bond between the two molecules. This oxygen bond is called glycosidic linkage. In this process, the alpha hydroxyl group of the alpha D glucose residue reacts with the carbon 4 hydroxyl of the second residue with the elimination of the water molecule to give disaccharides. So, how disaccharides are formed? When two monosaccharides, alpha D, page number 41, you can see the disaccharides. When the alpha D glucopyranose and two molecules of alpha D glucopyranose, the C1 and C4 of the H and OH molecule, water molecule eliminate and it forms the maltose. The two molecules join together with a oxygen link between the two molecules. This oxygen linkage is called glycosidic linkage. The linkage formed between the two monosaccharides is called that glycosidic linkage, represents 1,4 as alpha 1,4 glycosidic linkage. This page number 42, see. This nomenclature highlights the fact that linkage between carbon 1 atom of the first residue and the carbon 4 of the second residue, and secondly alpha anomer of the first residue involved. So this is the how alpha D glucopyranose and beta D glucopyranose, two molecules of beta D glucopyranose on hydrolysis, the two water molecule eliminates, it forms the cellulose disaccharides. Then polysaccharide. How polysaccharides are formed? Polysaccharides, these are called homopolysaccharides and heteropolysaccharides respectively. Polysaccharides chains may be linear, polysaccharide linear, it can be linear or branch chain. These are generally unreactive as most of their hemiacetal group are in the glycosidic bond. These polysaccharides are formed as many monosaccharides units are joined together. 10 molecules of the maximum 10 molecules of the monosaccharides join together to give the polysaccharides. And how they join? Na? Hydrolysis with the glycosidic bond. What is glycosidic bond? Na gote oxygen. One oxygen, the two molecules join together by oxygen linkage. Then what is starch? It is the carbohydrate reserve form of a plant which is a glucose polymer. Glucose polymer is called starch. In case of shortage of glucose in the cell, starch is hydrolyzed within the cell to release the glucose. Dietary starch is present in potato, rice, wheat, corn and many other plant sources. Starch is composed of two constituents, namely amylose, a polysaccharide with linear chains is made up of D-glucose units, and amylopectin. Amylopectin is a polysaccharide which is made main chain that are identical to those of the amylose with side chains joined by glycosidic linkage. Then glycogen. It is the storage form of the glucose in animals and is mainly concentrated in the liver and muscles. Its structure is the same as that of the amylopectin. However, glycogen is the branched to a greater degree and therefore contains 1 to 6 glycosidic linkage. In the glycogen, maximum 6 number of glycosidic linkage are present. What is glycosidic linkage? As I told, glycosidic linkage is a molecules are joined together by through a oxygen bond. This is called the in the glycogen 6 glycosidic linkage are there. That means 6 oxygens, 6 carbohydrates are joined together by oxygen through oxygen bonds. Then cellulose. It is mainly responsible for the structure of cell wall of the plants and consist of long chain made up of the D-glucose units joined by beta 1,4 glycosidic bond. These chains are closely attached to one another by hydrogen or van der Waals type of bond, thus forming compact and insoluble fibrous structure which permit the industrial use of the cellulose in the paper fiber and fabrics. Then what is glycoconjugates? Oligosaccharides and polysaccharides. As you know that oligosaccharides are number of carbohydrates are joined together to form oligosaccharides. Polysaccharide maximum 10 number of carbohydrates joins to give the polysaccharides. These, these oligosaccharides and polysaccharides these oligosaccharides and polysaccharides also exist in covalent linkage with lipids and proteins to form complex known as the glycoconjugates. Glycoconjugates are divided into broad classes: glycoprotein, glycolipids, glycosaminoglycans, and prote- proteoglycans. Let us discuss the what is glycoprotein. Many proteins in the cell are covalently associated with the carbohydrate molecule, resulting in a type of biomolecule known as the glycoprotein. The carbohydrate content such glycoprotein varies from 1% up to 85%. These molecules occur in almost all form of life, include most of the plasma protein, enzymes, hormones, etc. Then glycolipids. Glycolipids are the membrane lipids found in animals and plants in which oligosaccharides are attached. These oligosaccharides act as the cell surface markers by providing specific sites of recognition by carbohydrate binding protein. Glycolipids are widely distributed in most tissue of the body and particularly in the nervous tissue. Glycolipids are present in the nervous tissue such as brain, the plasma membrane. These contribute to cell surface carbohydrate which function cell membrane receptors are involved in the recognition membrane. Example: galacto galactocerebroside or galactosyl ceramide is given below page number 46. The structure is the structure of the galactocerebroside. Galactocerebroside has the characteristics 24 carbon fatty acids as constituent part, and it is also it is their galacto unit and galactose unit, monogalactosyl, galactosyl diglyceride, and sulfoquinovosyl diacetylglycerol. Blood in the page number 47, you can see. Then glycosamine are made up of alternate glucuronic acid and hexosamine residues. Then lipid. What is lipids? Lipids are a group of naturally occurring organic substances that are soluble in the organic solvent. Lipids are dissolved in the organic solvent such as the chloroform, benzene, etc., and highly hydrophobic. And hydrophobic means it is not soluble in the water and thus water insoluble. Most of the lipids are derivative of fatty acids. Lipids can be classified as their structure and function. They are explained. Simple lipids. What is simple lipids? Simple lipids are esters of fatty acids with alcohol, glycerol, oil, and fats are common example of this class of lipids. Fats are generally esters of the fatty acids with glycerol, and in fat, the liquid state is known as the oil. Wax are also simple lipids and are esters of fatty acids with high molecular weight, monohydric alcohol. Then complex lipids are also esters of fatty acids with alcohol. So lipids are classified as simple lipid, fatty acids, waxes, complex lipids. The group contains the following type of lipids. What is complex lipid? Now, phospholipids. Phospholipids are the glycerophospholipids. These are complex lipids, contains phosphate group. Phospholipids. It contains phosphate group. Glycolipids contains carbohydrate group, and other complex lipids are uh, derived lipids, storage lipids, membrane lipids, and specialized lipids. Then fatty acid. What is fatty acids? Fatty acids are the characteristic component of the lipids present in both animal and vegetable fats, such as ghee, coconut oil, and mustard oil. Fatty acids are generally named after the parent hydrocarbon. Fatty acid, fatty acids. The parent hydrocarbon chain contains no double bond. In fatty acid, there are no double bond, is unbranched, therefore I'll call the saturated fatty acids. In some fatty acid, the chain contains one or more double bond. It is called unsaturated fatty acids. So fatty acids, example oleic acids and octa and decanoic acids. These are the fatty acids. These are called the unsaturated fatty acid, which contains double bond, and saturated fatty acids, which does not contain any double bonds. The unsaturated fatty acids containing one double bond is called monounsaturated fatty acids. Those fatty acids having two or more double bond is known as polyunsaturated double bonds. Essential fatty acids. These have to supplied through the dietary sources, being essential for normal health and well-being of the organism. Such fatty acids known as the essential fatty acids. The classification on the basis of presence or absence of double bond can be represented by the scheme. Where fatty acid where the double bonds are present or not, they are divided as the saturated, unsaturated. Saturated hela animal fats are rich in saturated acid. Unsaturated vegetable fats. Vegetable fats are rich in unsaturated acid. Then unsaturated acids are divided in two groups, monounsaturated and polyunsaturated. Monounsaturated, one double bond is present. Polyunsaturated, more than one double bonds are present. Then triacyl glycerol. The esters of the fatty acids are acyl glycerols, and tri-substituted derivative being neutral in nature are called triglycerides or triacyl glycerols. The molecules constitute the fat. Fats are major form of the simple lipids. As mentioned above, there are fats are known as the glyceryl esters. The esters groups are glycerol. When treated with the ester, it gives the monoacyl glycerol. Then when another molecule of ester is joined, react with the monoacyl glycerol, it gives diacyl glycerol. When another molecule of ester reacts with the diacyl glycerol, it gives triacyl glycerol. Triacyl glycerols from animal sources normally contain higher percentage of saturated fatty acids and solids at room temperature. These are generally known as fats. With those, the vegetable sources are rich in unsaturated fatty acids and generally liquid at room temperature. These are known as the oils. Pure oils and fats are colorless, odorless, tasteless. When triacyl glycerol is subjected to hydrolysis, the ester bonds are hydrolyzed. Okay, the triacyl glycerol, the three ester molecules are react with the glycerol, and when the hydrolysis, the ester bond, it is general when ester is there, when hydrolysis, ester bonds are hydrolyzed. It gives consequently three molecules of fatty acids and one molecule of glycerols are obtained. These reactions can be catalyzed by acid, alkali. Alkaline hydrolysis of fat is called saponification. Since the fatty acids are liberated, would be present as their salt, which are soaps. An oil or fat can be characterized by the saponification number, which is the weight of potassium hydroxide in milligram required to completely saponify 1 gram of fat or oil. The iodine number of a fat is defined as the weight of iodine in grams that can taken up by 100 grams of fat or oil. A higher iodine number indicates greater degree of unsaturation in the fat. Higher is the iodine number, you can know the unsaturation is more. That means double bonds are more in that compounds. Then phospholipids. Phospholipids are the major lipid component of all biological membranes. These are so named. These are so named because of the presence of a phosphate group. Phospholipid. There are phosphate groups present. In addition, glycerol, fatty acids, and nitrogenous bases are the key components of this molecule. Hence, these are called phosphoacyl glycerol, also called phosphoglycerides. These are similar to triglycerols except that a terminal OH group. Okay. The phospholipids are classified on the basis of the nature of the polar substituted alcoholic group. These are phosphatidic acid and phosphatidyl glycerol, phosphatidyl choline, phosphatidyl ethanolamine, phosphatidyl inositol are like this. Then lipoprotein. Lipoprotein, what are the lipoprotein? Lipids being insoluble in water cannot be transported in the free form in the circulating blood plasma. Hence, these are transported in associated with the proteins. Such are called lipoproteins. Lipoproteins. Then the what is cholesterol actually? Cholesterol. One of the most common steroid is cholesterol, which is widely distributed in the body cell and is a major constituent of the plasma membrane and plasma lipoproteins. Terpenes are another large group of the lipids, which are made up of repeating isoterpenoids units are the precursor of the steroid. Terpene is a steroid. Terpene molecules are derived by condensation of the isoprene unit. Isoprene unit is 1,3-butadiene unit actually, which gives rise to such important components of the carotenoids from which vitamin A is derived, and cholesterol from which several steroid hormones, bile acid, vitamin D derived. Bile is a yellowish brown or green fluid produced in the liver and stored in the gallbladder. Bile, yellowish liquid present in the liver and stored in the gallbladder. Bile salts like the soap and other emulsifiers. This contains both polar and non-polar region, helping break down the fats in the foods into smaller pieces so that food is hydrolyzed more easily. Then steroid hormones. Steroid hormones, there are a variety of steroid hormones are there. Hormones are chemical released by cells and glands in one of the part of the body. They send out message. Hormones send out message that affect the cell in other parts of the body. Many hormones are steroid. Steroid hormones can be divided into 5 classes: glucocorticoids, mineralocorticoids, androgens, estrogens, androgestins, glucocorticoids, and are synthesized in the adrenal gland are collectively known as adrenal corticosteroids. Structure of estrogen in the page number 58, you can see the structure of the glucocorticoids, structure of the aldosterones, structure of the estrogens are there. Estradiol and estrogens are female sex hormones known as the estrogens. These are secreted by the ovaries and are responsible for the development of the female secondary sex characteristics. These are regulate the menstrual cycles. Progestogen is the hormone that prepare the lining of the uterus for the implantation of a fertilized ovum and is essential for maintenance of the pregnancy. And then eicosanoids. The polyunsaturated fatty acids are essential for animal and human nutrition because these are precursor to a class of compounds collectively called eicosanoids. Lipid aggregates. Lipid aggregates, the monolayers are formed when few lipid molecules are added to aqueous solution. But it's micelles. Micelles are spherical structures that contain anywhere from a few dozen to a few thousands amphipathic molecules. These molecules are arranged with their hydrophobic tails aggregated in the interior. Bilayer is the another type of lipid aggregate in water in which two lipid monolayer are arranged back to back to form two-dimensional sheet as shown in the figure. Liposomes are the closed cell sealing solvent filled basically bounded by a single bilayer. Biological membrane. Then, as we know from this, the polyunsaturated fatty acids have a cholesterol lowering effect and are thus dietary means of preventing coronary heart disease. They are important structural component of the cell membrane and mitochondrial membrane. Cholesterol is the precursor molecule for synthesis of the bile acids. Lipids are a group of naturally occurring organic substance usually formed by combination of the fatty acids with glycerol. Lipids are soluble in organic solvents such as ether, chloroform, etc. Lipids are highly hydrophobic, that means not soluble in water. Lipids can be classified on the basis of their simple, complex, and derived types. Cellular organelles such as mitochondria, nucleus, lysosome, and chloroplast are bound by the membrane. The membrane gives individuality to the cells, separating them from the environment and permitting cellular function. The biological role of lipid include prevention of loss of heat, packing and support material, most concentrated form of stored energy, cholesterol lowering the effect, thus a dietary means of preventing coronary heart diseases, etc. So this contains, so these units we learn the what is monosaccharide, polysaccharide, disaccharide, lipids. Then coming to the unit 3, proteins and nucleic acids. Proteins and nucleic acids, amino acids which have either amino acid attached to the alpha carbon is known as the alpha amino acids. Alpha carbon carries a hydrogen, alpha carries a hydrogen atom. The fourth group on this carbon is known as the side chain and denoted as R. Alpha amino acid is example of alpha amino acid is glycine, has a hydrogen atom as the side chain. Alpha amino acid, this side chain consists long carbon chain and more complex chemical groups. Representation of the amino acids. The amino acid is commonly represented by 3-letter symbol, 1-letter symbol. Glycine represents the GLY. Glycine we can write by abbreviation name GLY. In addition to the above, 1-letter symbol is also in use. 1-letter symbol for even 11 of the 20 coded amino acid is the first letter of their full name. Classification of the amino acid. How many types of amino acids are there? It is there are aliphatic amino acid, hydroxy amino acid, sulfur containing amino acid, acidic amino acids, basic amino acids, aromatic amino acids, amino acids. Basic amino acids contains additional basic NH2 groups. Aromatic amino acids contains example tyrosine and tryptophan. So these are the classification of the amino acids. Classification of amino acid based on the polarity of the R group. Non-polar R group, polar uncharged R group, polar negatively charged R group, positively charged R groups are there. Then essential and non-essential acids. Our body can synthesize only 10 of the amino acids are called non-essential amino acids as indicated by asterisk. The rest are not synthesized in humans but are synthesized in either plant and bacteria. So non-essential amino acids our body can synthesize, but all essential amino acid essential amino acid, these essential are leucine, isoleucine, lysine, methionine, polyalanine, tryptophan, threonine, vanille, histidine, proteins that contains all the 10 essential amino acids are called adequate proteins. Milk and animal proteins are an excellent source of all amino acids, whereas plant protein lack one or more of the essential amino acids. However, protein in soybean is an exception and is adequate protein. Amino acid arginine and histidine belong to the semi-essential amino acid because body does not always require dietary sources for it. So these are the variety of the in the page number 74 you can see the structure of L amino acid with three and one letter name glycine, alanine, valine, leucine, isoleucine. These are the amino acid non-polar R groups are there and the proline, phenylalanine, methionine, tryptophan, serine, cysteine, threonine, asparagine, glutamine, tyrosine, aspartic acid, glutamic acid, histidine, arginine, lysine, all are these are essential amino acids. In the page number 74, their structures are there. Then characteristics of the amino acid. They contain both the acidic in amino acid. They both contain the acidic group and basic group. Acidic group contain the carboxylic group COOH group and also a basic group NH2 group. They an amino acid usually represented in its own ionized form. However, there are represent ionized form which predominates under the physiological condition. We have already mentioned that amino acid contain at least one carboxylic group and one amino group. Both the groups are capable of ionizable in an aqueous solution. Thus, in water, amino acid can act as both acids and bases. Molecules which exhibit this property are known as the amphoteric in nature. They are capable of ionization in aqueous solution. They act both acid and bases. These are called amphoteric molecule. That's why they are called amphoteric molecule. In other words, this means amphoteric molecule can accept as well as the donate a hydrogen atom. It acts as both acid and base. So it is amphoteric in nature. uh, what is isoelectric point? We have already the Jupiter ion have one positive and negative charge and are electrically neutral. Therefore, these dipolar ion don't migrate in an electric field. The pH at which amino acid carry no charge is called isoelectric point. There is a point, the pH, where that amino acids carry no charge. No charge, that means the pH at which amino acids carry no charge is called isoelectric point. For example, isoelectric point for glycine pH is 6.0, which means that at this pH, glycine will be Jupiter ionic form. Depending on the pH of the aqueous solution, amino acids can exist in the different ionic form. As it contains both acidic and basic group, it exists in the different ionic form. Amino acids earlier separated from the mixture of amino acids are obtained by hydrolysis of plants and animal proteins. Then formation of the peptide. How peptides are obtained? Two or more amino acids react together to form peptide. Peptides are formed from the amino acids. Two or more amino acids, page number 79. Two or more amino acids react together to form the peptide by elimination of the water molecule. When water molecule goes out, it forms the peptide bond. Amino acids that contain together can be same or different. We may call it as a condensation reaction as it proceeds with the elimination of a water molecule. The reaction between the alpha carboxylic acid of one amino acid, alpha amino group of the other amino acid with the formation of the amide linkage. As we know, the amino acid contains both the NH2 group and COH group. So COH group one uh, between alpha carboxylic group one amino acid, alpha amino group of the other amino acid with the formation of the amide linkage. Amide means CONH. Amide linkage is there. Two molecules of the amino acid joined together by amide linkage. The amide linkage are called the peptide bonds. the amino acid, two molecules of the amino acid, one amino acid CO acid group and other amino acid group NH2 group joined together, it form amino acid resid peptide, it form a peptide, and by attaching a CONH joined through a amide bond, that is CONH bond. The above reaction can continue, page number 80. You can see the amino acid, two amino acid, it form two amino residue is there, in the middle CONH amide linkage is there with loss of the water molecule. So glycine and alanine, it form dipeptide alanine. Structure of the peptide bond and nomenclature and representation of the peptide. You should also note that the peptide is named from the amino end, which is having free or acylated alpha amino group. For example, the tetrapeptide given earlier, this amino acid ends, it's called N terminal is glycyl residue and C terminals. There is a N terminal group and C terminal group joined together. Representation of the peptide group. Its structure is there in the page number 83. And the classification of the protein, the type of protein can be classified based on their solubility, shape and structure. Fibrous proteins are formed by the polypeptide chains and appear as the long filaments. Globular protein appear as the it's called the spheroproteins and corpuscular protein. Examples are insulin, albumin, globin, simple protein and conjugated protein. Conjugated protein or heteroproteins contains in their structure and non-protein part of the group. Derived proteins are also proteins derived by the action of different physical and chemical agents like heat, acid, alkali, enzyme. Biological functions of the protein. Biological variety of function that protein perform the consequence of both folding and polypeptide chain. Structural protein, enzymes, regulatory proteins, transport proteins, storage proteins, protective or defense proteins. Structural organization of the proteins, primary structure of the protein. As mentioned, a protein is assembled as a linear long chain polymer of the amino acid is called polypeptide. Number and sequence of amino acid residue from N terminal to the carbon terminal, which is linked linearly by peptide bond, is referred as the primary structure of a protein molecule. Peptide bond and protein folding. The folding pattern of the polypeptide chain depend on the geometry of the peptide bond. Disulfide bond. Disulfide bond, a bridge, are extremely important in stabilizing higher order protein structures. This bond may connect two polypeptide chains. Disulfide, the two molecules of sulfur atom joined. Now, polypeptide chain, two polypeptide chains joined by the two sulfur atom, it called disulfide bond. Secondary structure of the protein. Secondary structure, extensive investigation by Pauling and Corey using X-ray diffraction studies. They give the structure of the protein. They are ordered and stable elements of protein folding is called secondary protein. Secondary structure is basically the specific geometric arrangement of the amino acid. Geometric arrangement of the amino acid that results from amide linkage that are close to each other in the polypeptide chain of a protein. They have the alpha helix. Secondary structure of the protein have the alpha helix structure. Alpha helical structure results from a particular pattern of rotation around the nitrogen alpha carbon and alpha carbon carbon bond of the polypeptide chain, which means polypeptide chain can turn back under itself in a spinal or alpha helix. Alpha helix, thus a tightly coiled structure that resembles a right-handed screw or a circular staircase. This arrangement allows hydrogen bonds to form between the carboxyl group of the 1 amino acid and NH group of the 4 amino acid. The beta plated sheets in this arrangement Polypeptide chains are almost fully extended and hydrogen bonding occur between the close zigzagging parallel chains. The tertiary structure, the primary structure, secondary structure, and tertiary structure of the protein. In most of the large number of the protein include enzyme, polypeptide chain is twisted, folded, packed in compact, almost globular three-dimensional shape. Tertiary structure of the protein, three-dimensional shape, which is known as the tertiary structure. In this tertiary structure, stretches of the secondary structure, which alpha helix and beta sheets, along with the randomly coiled region, those without any particular order of the polypeptide chain, fold back on each other in the three-dimensional, forming compact globular shape structures. The folding of the entire, these are the folded structure. Folded proteins are the folding type of structure. The folding of the entire protein molecule, the tertiary structure is stabilized by hydrogen bonding and hydrophobic interaction, which are principally responsible for the specificity of the folding. However, other interactions such as the van der Waals dispersion forces to make a significant contribution because amino acid side chains are closely packed in globular proteins. A small contribution to the stability of the folded structure also comes from electrostatic forces like interaction between the carboxylate group of one amino acid and NH3 plus group of the another. Disulfide bonds are also provide further stability and the quaternary structure of the protein. In this level, tertiary structure of the protein, tertiary, then quaternary structure of the protein. Quaternary, this level of protein organization involves association of the two or more individual protein unit. Each tertiary structure into complex and functional unit. This association of various protein subunits is also known as the quaternary structure. The relationship between the tertiary and quaternary structure can be best you can see in the myoglobin and hemoglobin. Hemoglobin and myoglobin structures are there in the page number 95. Myoglobin function as a oxygen storage protein in the muscles, whereas hemoglobin transport oxygen throughout the body. Myoglobin binds oxygen when oxygen content in the cell is high and releases it when the level in the cell falls. You will recall that myoglobin has tertiary level protein organization and we have already represented its structure. A non-protein molecule called heme with an iron atom in the center in the oxygen binding site is the myoglobin molecule. Myoglobin heme protein is there. It is located in the nonpolar cavity of the myoglobin molecule and bonded to a histidine molecule in the chain. Hemoglobin is present in the red blood cell where it binds oxygen and then transport it to the tissue throughout the body. Then nucleic acids. Nucleic acids as means nucleotides, the building block of nucleic acid. The polynucleotide are the building blocks of nucleic acid in which the nucleotide units are joined by phosphodiester linkage. A nucleotide contains phosphate and nitrogen containing base. There are 5 bases in a nucleotide. Nucleotide 5 bases are there. 2 are substituted purines are called purine base, adenine, guanine, and 3 are substituted pyrimidines. Pyrimidines bases are cytosine, thiamine, uracil. So nucleic acid, polymer of the nucleotide, when it reacts with the water catalyst, it forms a nucleotide. RNA and DNA have certain common constituents. These are the phosphate groups, the purine base, namely adenine A and guanine G, and pyrimidine base and cytosine C. However, the major difference between RNA DNA is the former contains the pentose sugar ribose, where the DNA contains 2-deoxyribose as the pentose sugar. Beside the bases, uracil is found only in the RNA and base thiamine present in the DNA. So adenine, guanine, thymine, uracil, cytosine, all the structures are present. All the structures is given in the page number 99. These are the RNA and DNA. Nucleosides and what is nucleoside and nucleotide? When a nitrogen base, whether substituted purine or substituted pyrimidine, is covalently linked to a pentose sugar, the resulting molecule is the nucleoside. The deoxyadenosine, deoxyguanosine, deoxycytidine, thiamidine, and adenosine, guanine, cytosine, uridine. The structures are given in the page number 100. You can see when a phosphate group is attached to a nucleoside, the resulting molecule is known as the nucleotide. What is nucleotide? When the phosphate molecule is there attached, it is called nucleotide. The most common site of the formation of the phosphodiester linkage is the 5 and 3 position of the ribose or deoxyribose molecule. Mm. Okay. Then the deoxyribose acid, deoxyribonucleic acid, DNA is deoxyribonucleic acid, is the polymer of the deoxyribonucleotide with a structure similar to that of the RNA, except that pentose unit is 2-deoxyribose. DNA is the fundamental unit of heredity. DNA has a pronounced secondary structure consisting of two independent polydeoxyribonucleotide strands coiled around a common axis and associated together by hydrogen bond. Ribonucleic acid, ribonucleic acid, RNA are polymers of the ribonucleotide having 5-3 polyphosphodiester linkage. RNA is almost always have the linear structure. The RNAs, in fact, are the middleman of the informational transfer, depending upon the different role they play in the biosynthesis of protein and polypeptide. RNA, ribosomal, these are the most abundant RNA and are present in the ribosomes in a cell. The ribosomal RNA account for 60% of the total RNA, which indicates their important structural role. Messenger RNA, mRNA. Messenger RNAs are much shorter than the ribosomal RNA and are the active RNA in the polypeptide synthesis. Transfer RNA, tRNA, transfer RNA. Transfer RNA takes part in the amino acid and transport system in the cell that carries the individual amino acid to the site of the synthesis of the protein. So, in the summary, DNA, we know that DNA is a universal genetic material. All form of the life owe their origin and existence to them. The DNA contains four hydrogen nitrogenous bases, two purines, adenine, guanine, and two pyrimidines, uh, cytosine, thiamine. Pentose sugar in the DNA is deoxyribose, and ribonucleic acids are the other nucleic acid consisting of nitrogenous bases, adenine, guanine, cytosine, uracil, ribonucleic acids. RNA, uh, RNA, the base uracil replaces thiamine, which is present only in the DNA. There are three kinds of RNA depending upon the role played by them in the polypeptide synthesis. These are ribosomal RNA, messenger RNA, and transfer RNA. So we learned what is DNA and what is RNA, what is messenger RNA, what is the transfer RNA. So coming to the uh, unit 4. In this unit, we learn the define the metabolism and differentiate between the catabolism and anabolism. Describe the general design of the metabolism. It defines the bioenergetics and describe the significance, formation, and utilization of the ATP in the living system. It explains the maintenance of water balance in the body. So, overview of the metabolic process. Metabolism is the sum of all enzyme and catalyzed chemical reactions taking place in an organism. The word metabolism is derived from the Greek word metabol, meaning change. It is also called intermediary metabolism as a substrate goes through the multiple intermediary steps to form an end product. Outcome of this, the breakdown of the complex biomolecules obtained from the environment into simple usable compounds, the efficient extraction of the chemical energy of the biomolecules into ATP and reducing equivalent for biosynthesis and other cellular activities, synthesis of complex biomolecules from simple precursors in accordance with the changing needs of the organism, synthesis and storage of long and short term energy reservoirs in condition of the excess. Metabolic breakdown of the carbohydrate, fat, protein shows that these may be considered to proceed in the three stages. In the first stage, large nutrient molecules are hydrolyzed to smaller nuclear molecules, which are the building blocks. So, what is metabolic process? The breakdown of the food, carbohydrates, fats, proteins, metabolic breakdown. A limited amount of energy is released in this stage. When the it break down, a limited amount of energy is released in this stage, part of which is utilized to drive the net synthesis of a small number of ATP molecules. So, these two stages can proceed anaerobically. In the third stage, the common products obtained in the previous stage are oxidized to the carbon dioxide and water by a cyclic process called a tricarboxylic acid or citric acid. Catabolism and anabolism. What is catabolism and anabolism? Catabolism is a Greek word refers to the reaction involved breakdown of the complex biomolecule. Breakdown of the complex biomolecules such as carbohydrates, lipids, proteins, simple molecules such as carbon dioxide, NH3, ammonia, and water. These reactions are energy generating and oxidative in nature. Some part of the energy released is utilized for the synthesis of ATP or reduced energy carrier NADH, NADPH, and FADH2, and the rest is lost as heat. Uh, a less apparent role of catabolism is to provide a variety of anabolic precursors. Finally, catabolic sequences are convergent in nature. The TCA cycle is a convergent cycle for the complex oxidation of the carbon. Anabolism, on the other hand, involves step-by-step biosynthesis, simple and complex biomolecules. Ana- Anabolic pathways are divergent, catabolic pathways are convergent and depend on a source of energy that is made available. Convergent nature of catabolism of fats, carbohydrates, proteins, which give rise to some common metabolites. Biosynthesis of these classes compound proceeds mostly but not entirely. Anabolism is divergent in nature and since that diverse set of compounds, namely carbohydrate, fat, and proteins, are synthesized from a common set of precursors. Design of metabolism. The reactions that occur in all organisms are in very large numbers. Even simplest unicellular organism is capable of catalyzing a few thousand reactions. A unique attribute of life is its ability to regulate metabolic process. This providing both flexibility and economy, which means that the organism can adapt to changing needs and express the appropriate enzymes as when they are required. Introduction to bioenergetics. What is bioenergetics? You know that cell has a well-defined structure built from complex organic molecule. Cell is a well-defined structure built from the complex organic molecules. It is made possible. This is made possible by continuous flux of energy through the living system and its transformations for various processes. Study of such transformations comes under the purview of bioenergetics, which include generation of energy by the breakdown of the nutrients. Generation of energy by breakdown of nutrients, its storage and utilization in performing various functions of the cell. Further, biogenetics attempts to unravel the complex network of biochemical pathways involving a large number of interferent biochemical reactions. We are sure that we have studied the laws of thermodynamics. As you have the knowledge of the thermodynamics, a significance conversion of this principle as used in the biochemical reaction have a few difference which you will study in the following section. Convention in the biochemical energetic. Biochemical reaction, two conventions are very important. First one concern the standard state and the standard free energy change. Del G 0 equal to del G 0. Hence, the proper care should be uh, additivity of del G value and coupling reaction. A to B reaction, del G free energy is equal to GB minus GA. Finally, an important corollary of the additively free energy value is that certain unfavorable reactions can be made to proceed by suitably coupling these with highly favorable reactions. When ATP overall reaction ATP plus glucose, it gives glucose 6 phosphate plus ADP. So this is the reaction plus some free energy del G0 is equal to minus 17.2 kilojoule per mole. Mode of coupling is essentially a manifestation of law of mass action. In this above reaction, glucose when treated, the reaction when glucose treated with the ATP, added with ATP, it gives glucose 6-phosphate. This reaction would not couple if they are occurring independently. This reaction is catalyzed by enzyme is called hexokinase, which facilitates the transfer of the phosphate group from the ATP to the glucose. Okay, when glucose treated with the ATP, the phosphate group added to the glucose moiety and the phosphate group comes from the ATP molecule. The favorable reaction either proceeds or follows the unfavorable reaction. If it proceeds, it provides the reactant for unfavorable reaction a large amount and push it ahead. And the hydrolysis of ATP, the biological energy carrier. Uh, Adenosine triphosphate is a adenosine triphosphate ATP. ATP. Adenosine triphosphate is a
ମ୍ୟାମ୍
nucleotide.
ଟିକେ
But
ଫେଭରେବୁଲ ଆଣ୍ଡ ଅନଫେଭରେବୁଲ ଟା ଟିକେ ଆଉଥରେ କହିଦେବେ, ବୁଝିହେଲାନି।
ହଁ କଣ ହେଲା କହିଲେ?
ଫେଭରେବୁଲ ଆଣ୍ଡ ଅନଫେଭରେବୁଲ କଣ୍ଡିସନ ଯୋଉଟା କହିଥିଲେ ଯୋଉ ଡେଲଜି କୁ ନେଇକି ସେଇଟା ଟିକେ ବୁଝିହେଲାନି, ଆଉଥରେ ଟିକେ କହିଦେବେ।
ହଁ ଡେଲଜି। Mhm. Okay. Mm, the value of del G depend upon the initial and final state of the system. When a system is transferred from the state A to state B, state A ରୁ state B, del G ହେଲା GB minus GA. State A ରୁ state B କୁ ଗଲା GB minus GA. Okay. This change may taken place in the single step. Okay. Single step रे it transfer from A to B. That means alternatively, the reaction may take place through an intermediate. Okay. Okay. A to B, it can directly go to A. It is not directly going to A to B. It comes A to B through a intermediate, that is I. A to I. A ru I ki gala. That means del G hela I minus A. I to A. A ru I ki gala. I minus A lekha ହେଇଛି. ତାପରେ I ରୁ B କୁ ଗଲା. Then I to I is the intermediate step. Then I to B. That means del G is equal to B minus del G B minus I. It's written. The single del G depends upon the initial and final state only. So del G B A to B equal to del G I A plus del G B minus I. Can you understand this one?
ଇଏ ସେମ୍ ବଟ୍ ଇଣ୍ଟରମିଡିଏଟ ଷ୍ଟେଟ୍ ର ଏଇଠି କାମ କଣ ରହିଲା ସେଇଟା ଟିକେ ବୁଝିହେଲାନି। ମାନେ ଇନିସିଆଲ ଆଣ୍ଡ ଫାଇନାଲ ଷ୍ଟେଟ୍ସ ଉପରେ ତ del G ଡିପେଣ୍ଡ କଲା ନା।
this change may take place. Okay, it may not happen that directly A to B reaction is not happening. It may take place. Like he say, a reaction it may take place ଗୋଟେ intermediate step ଦେଇକିରି ହିଁ ଯାଉଥିବ. The
ଆଜ୍ଞା
reaction may takes place through the intermediate. ତେଣୁ
ଆଜ୍ଞା
del G B to A, it del G I minus A plus del G B minus I. Okay, these two
ଆଜ୍ଞା
step mixing and del G A to B the reaction. Okay.
ଆଜ୍ଞା
In other words, thermodynamic equation can be added or subtracted like algebraic equation. This is a very useful relationship. It helps us in calculating del G or del G0 or del G0 value for reaction where direct and determination may not be conventionally carried out. Uh, these are highly favorable reaction. Coupling of the reaction can take place in the different way. One of these, glucose PI, glucose 6%. Then I told when the glucose react with ATP, it gives glucose 6 phosphate plus ATP. Glucose 6 phosphate group comes from your ATP group. For an unfavorable reaction, ATP glucose 650. It may, however, be noted that above two reactions would not couple if they are occurring independently. Mm, Okay, this reaction is catalyzed by enzyme is called a hexokine. Oh, this is in this reaction one enzyme they are using hexokinase, which facilitate the transfer of phosphate group from the ATP to glucose. Without enzyme, this reaction will not happen. That enzyme helps to transfer the phosphate group from the ATP group to the glucose group. Uh, this favorable reaction either proceeds or follows the unfavorable reaction. If it proceeds, it provides the reactant for the unfavorable reaction in large amount to push it ahead. If it allows the unfavorable reaction, it continuously removes the product of first reaction, thereby effectively pushing the reaction through the large amount to push it ahead. For an unfavorable reaction, B to C, highly favorable reaction like A to B or C to D can act as suitable coupling reaction where the former would have to proceed and latter would have to follow the reaction B to C. For example, acetyl coenzyme plus oxaloacetate enzyme A, it gives citral coenzyme B citrate plus coenzyme. The del G0 for step A is close to 0, uh, while the step B is highly exerogenic and overall reaction quite favorable and proceed in the right direction to a good extent. The energy carrier in the living system for various energy requiring process is adenosine triphosphate, about which you will study in the next section. Okay.
ମ୍ୟାମ୍ del G ଜିରୋଟା ଜିରୋ ହେଲା ବଟ୍ ନେଗେଟିଭ ବି ଲେଖେଇଛି।
And del G0
Step A
negatively.
ରେ ଲେଖିନି del G0 in for step A is close to zero but negative।
Close to 0 but negative.
ଆଉ exergonic ମାନେ ମ୍ୟାଡାମ କଣ exergonic ମାନେ?
Energy ତା ମାନେ energy released ହଉଛି।
ଆଜ୍ଞା 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 ଓହୋ।
ହଁ। Then hydrolysis of ATP. ତ biology ଜିନିଷ ଗୁଡା ଦେଇଛି। Are you chemistry student? Are you
Yes ma'am,
chemistry or biology? Chemistry or biology?
chemistry।
Chemistry. BSc chemistry.
Yes ma'am।
Okay. So many biology things are here. Okay. The hydrolysis of ATP. You can see ATP, adenosine triphosphate. ATP is a nucleotide consisting of a purine base, adenine. I am also a chemistry background actually,
ଓହୋ ଆଚ୍ଛା।
chemistry background. I told them
ନାଇଁ
that
କେମିଷ୍ଟ୍ରି ବ୍ୟାକଗ୍ରାଉଣ୍ଡ କେମିତି ବାୟୋଲୋଜି କେମିତି?
yes, I told them that I would like to. I am a chemist. Why you are giving me so biology things are there? They told that already. The next time we'll change and this time please. You uh,
Mhm.
take the class and next time will change. Actually, I am pure organic chemistry. Mm. So I told them to change, but they did not change. Mm. Adenosine triphosphate is a nucleotide consisting of purine base, adenine, a ribose sugar, and triphosphate unit. Figure 5.2. 5. 4.2. 4.2. Structure of ATP and figure 4.2 gives the hydrolysis product. When ATP hydrolyzed, it gives ADP. Mm, ADP gives AMP. AMP gives adenosine, and ATP directly gives AMP. AMP. Okay, kill. Mm, Triphosphate gives structure of the ATP. It gives it hydrolyzed product, namely adenosine diphosphate ADP, adenosine monophosphate AMP, and the inorganic phosphate. A large amount of free energy is generated on the hydrolysis of the ATP as detailed in the figure. A large amount of free energy is generated. Free energy for the hydrolysis of ATP to ADP. The actual free energy change del G for the hydrolysis of ATP to ADP, and phosphate ion in a living cell is considerably different from given in the figure 4.2 and depends on the intracellular concentration of the reaction partner and are far from the unity and are unequal. Okay. Del G is equal to del G 0 plus RT log এটা হচ্ছে reaction del G is equal to del G 0 plus R equal to 2.303 RT log ATP PI ATP del
ପ୍ରଡକ୍ଟ
G
ବାଇ
0
ରିଆକ୍ଟାଣ୍ଟ ଅଛି ଆଉ।
हाँ del G 0 ହେଲା -30.5 प्लस एटा होला 2.303 आर तो होला 8.314 आका वैल्यू दे देला लगे ये करला एटा होला एतिक del G ହେଲା 51.8 -51.8 kilojule per mole. The numbers pertaining to del G 0 given above will be useful when we try to rationalize the choice of ATP as energy carrier. Okay. ATP ରୁ ATP ଟା ହାଇଡ୍ରୋଲାଇଜ ହେଇକିରି ଆମର ହେଲା ADP ପ୍ଲସ୍ some energy -30.5 kilojule per mole এটা ଦେଇଛି ରିଲିଜ ହେଇଛି ADP ADP then converted to AMP AMP hydrolyze AMP କୁ hydrolyze କଲେ এটা হচ্ছে adenosine. Then role of ATP in the biochemical energy transformation. ATP is continuously formed and consumed in the living system. In living system, ATP is continuously formed, act as energy coinage or energy currency of the living cell. Analogy of the role of ATP in the living cell is seen in exchange of currency. When you visit a foreign country, in that country you have to first get your money changed into a local currency in order to be able to pay for various services and merchandise. Similarly, all energy received by the living cell is converted to its own energy currency, ATP, before it can be utilized to drive the various energy requiring function. Role of ATP as an energy transducing agent or vehicle suggests that it should be rapidly turned over, that is rapidly and repeatedly broken down to ADP, to PI and resynthesized in the living cell. Okay, so ATP rapidly, repeatedly broken down to ADP and resynthesized in the living cell. An idea of the rapid turnover of ATP can be heard of from the fact that a resting adult human being turn over 30 to 40 kg ATP in the one day, that converts it into ATP and PI and then back to ATP. The relative concentration of the ATP, ADP and PI remain constant. Okay, so ATP in our body, ATP is repeatedly break down to ADP and again resynthesized in the living cells. When your glucose treated with the oxidized glucose Before we go to the chemistry ATP ADP interconversion, it is important to compare the free energy change involved in the process that obtainable from the oxidation of the common nutrient. When you see large amount of free energy become available in complete oxidation of the glucose C6H12O6 oxidation ହେଇଗଲା, oxidation which gives 6 carbon dioxide, water and some free energy, large amount of free energy available for an efficient utilization of this energy for synthesis of the ATP. This energy for synthesis of ATP, it is necessary that it should be liberated in the several smaller packets, each of which should be adequate to drive the synthesis of ATP. An analog situation occurs in electricity generation from the large waterfall when only available turbine is of small and limited capacity. Example this. Diagrammatic representation of the generation of electricity by large power fall. Oxidation of the glucose must take place by large number of the steps and therefore involve equal number of intermediates called metabolites. It is indeed found to be so and so common feature of all metabolic pathway. Therefore, metabolism is commonly referred to as the intermediary metabolism. Some example of metabolic pathway will be discussed in the unit 5-6. Structural basic role of ATP. Uh, ATP standard free energy and hydrolysis of some compounds of biological interest. Okay. Del G0 value for the hydrolysis of ATP is much larger than that of AMP. In the fact, it is in fact found to be larger than that of the hydrolysis of all simple phosphate ester. You see that phosphate, the uh, phosphate ester, glycerol 1 phosphate, glycerol 1 phosphate when did hydrolysis in water reaction ହେଇକି glycerol ହେଇଗଲା plus HPO4 minus 2 etc. This reaction hydrolysis of glycerol 1 phosphate and corresponding value del G0. Del G0 is minus 9.205, some energy released. Since del 0 value are related to the respective equilibrium constant of this reaction, it follows that ATP has a higher tendency. ATP has a higher tendency to transfer its terminal phosphate terminal phosphate group to water compared to glycerol 1 phosphate ester. Therefore, it is said that higher phosphate group transfer potential than the phosphate esters. ATP, ADP have been referred to as the high energy phosphate compounds and the phosphor anhydride and phosphate bond between the phosphate groups, and these compounds are often referred to as the high energy bond. There are several other high energy phosphate compounds who there are several other high energy phosphate compounds who participate in the metabolic process. The phosphate group transfer potentials of some of these are higher than that of the ATP. Some compounds other than phosphate derivatives also exhibit large del 0 value for hydrolysis. The purine base and ribose sugar of AMP, ADP, ATP contribute towards interaction of these with the enzymes. It is of importance in regulating enzyme activity. The above mentioned qualities, besides the stability of ATP, are responsible for its role. In the process of study mode of action of ATP, we have realized that ATP has to be rapidly turned over. Let us now see how this job is accomplished. Universal electron transfer. Cells contains free energy in a chemical form of catabolism of nutrient molecules, and they use that energy to make ATP from ATP inorganic phosphate. ATP then donates some of its chemical energy to endogenous process such as synthesis of metabolic intermediate and macromolecules from smaller precursor molecules, the transport of substance across the substance. Cellular oxidation of glucose to carbon dioxide requires specialized electron carrier. ATP synthesis, which is about 50 to 60 kilojoule per mole, cells converts glucose to carbon dioxide not in a single high energy releasing reaction but rather in a series of controlled reactions. Molecules that accept electron from electron donor and donate this to electron acceptor, creating an energy producing electron transport chain that occur in respiration and photosynthesis circle universal electron transfer. Aerobic organism ultimate electron acceptor in the oxidation of the fuel molecule is oxygen. However, electrons are not transferred directly to oxygen. Instead of fuel molecules, transfer electron to special carriers which are either pyrimidine nucleotide or flavins. All the cells in our body are surrounded by liquid called tissue fluids, which has exactly the right condition in which cells can work.
ମ୍ୟାମ୍ ଇଲେକ୍ଟ୍ରନ୍ ଟ୍ରାନ୍ସପୋର୍ଟ ଚେନ୍ ର କାହିଁ ଡାଇଗ୍ରାମ ହେରିକା କିଛି ଦେଇନି।
କିଛି ଦେଇନି କିଛି ଦେଇନି ଇଲେକ୍ଟ୍ରନ୍ ଟ୍ରାନ୍ସଫର ଚେନ୍ର। ଆକୁ କେମିତି ବୁଝିବ ତ ଯେ ଷ୍ଟ୍ରକ୍ଚର ଫକ୍ଚର କିଛି ନ ଦେଇଥିବ।
ହଁ
ତାକୁ ଅଲଗା ବହି ଫଲୋ କରି ଦେଖିବାକୁ ପଡିବ, ଜୁଲୋଜି ବହି ସବୁ। ଇଏ ଦେଇନି କିଛି। ତାପରେ what is homeostasis? Homeostatic control system and components. Homeostatic control system, it contains sensor, control center and effector. It is an important part of human body to maintain physical and chemical state at constant equilibrium by constantly monitoring the internal variables. Normal level of such variables in the body collectively referred as set points. In other words, the set point is the normal physiological value or range value necessary for body function. Homeostatic control center regulates set points, variable option, function of the body and life process. For example, our body has set point 70 to 100 milligram per dl for blood glucose, 37 degrees centigrade for body temperature at which our healthy body works the best. At the condition below or above this set point, body fails to work optimally. Okay, so there is some standard temperature where body work best. Jadi, if something changes, set point something, if condition below or above these set points, body fails to work. Some physiological factors such as body's temperature and the blood pressure through the tend to vary slightly below or above the normal range. However, the homeostatic controls system recognizes this, controls the system to recognizes this altered level of set points and sends information to the control center to release the chemical signal in order to regulate the variables in the body. So, homeostasis is the regulated by the four component mechanism. These are variables, receptors or sensor, control and effectors, and explain the brief. Stimulus, sensor, control center, effector. So they explain all one by one. Stimulus is the internal external variables. Set point of the body requires such variables stimulate the homeostatic control system through sensor or secondary receptor. Sensors are specialized chemical structures in organ which detect the change in the set points of variable. Example, beta cell of the pancreas detect the abnormal sugar level in the body. Receptor in the certain arteries detect the change in blood pressure, and sensor nervous cell monitor change in the body temperature. Receptor produce a signal and send impulse to the central center of action, control center. Control center are located in the brain which monitor the normal range of variables. For example, hypothalamus, small region in the brain is the thermoregulatory center of the body as it regulates the body temperature and maintains 37 degrees centigrade. When the body temperature altered, it receives information from sensory receptor, sends signal to the effector to stabilize the body temperature. Then effector, the cells, tissue, and organs that respond the instruction. That means, for example, sweat glands work as effectors by releasing sweat to lower body temperature in the body, and hepatocytes of the liver releases glucose to increase the blood glucose level. The feedback mechanism we explained in the following. That means hemostasis is it regulates certain body activities through all these system: stimulus, sensor, control center, and effector. The feedback mechanism. The what is feedback mechanism? Positive, negative feedback mechanism and positive feedback mechanism. Feedback mechanism कौन? So ये hemostasis रो negative. Most control system maintain hemostasis by negative feedback mechanism. It keeps the set point of the physiological state constant in the body. Enzymatic reactions are regulated by negative feedback systems. Mechanism homeostatic control system called the negative feedback shows regulation of the body temperature by negative feedback loop. In the increase in the body temperature act as the stimulus which is sensed by the nerve cells in the skin and brain. Sensors send the signal to the temperature regulatory center hypothalamus which finally sends signal to effectors to release the chemical messenger. Stimulus, sensor, control center, effector. The body temperature exceeds 37, nerve cell in the skin and brain, temperature regulatory center brain, sweat gland throughout the body temperature regulation. Negative feedback loop that maintain body temperature. Positive, what is positive? Positive feedback mechanism instead of reversing the change, increase the change in the body physiological condition. For example, during childbirth, the first contraction of the labor pushes the baby towards the cervix and stimulates the stretch receptor, receives signal and oxytonic hormone to release from the pituitary gland. Oxytocin causes stronger contraction of the smooth muscles, pushing the baby further down. This causes even greater stretching of the cervix, sending more signal. The cycle goes until the baby born. The cessation of the stretching of the cervix stops the release of the oxygen. Another example is the platelet aggregation and accumulation in the response to an injury. Positive feedback, negative feedback mechanism. Two mechanisms of the homeostasis to actually control the body temperature, maintaining water balance. The water balance in the human being in cool environment. Regulation of the water balance. Input of water as well as its loss can be highly variable due to individual habits and environmental factor. In spite of this, total body needs water to be maintained constant to achieve normal osmolality for physiological function. Osmolality is a measure of the osmoles of solute per kilogram of solvent. Osmol is a unit of measurement that defines number of moles of a chemical compound that contribute to solution osmotic pressure. Then homeostatic ରୁ ଆମେ ଜାଣିଲେ ଯେ homeostatic control is a control system is a signal directed mechanism which work in a feedback loop and can occur at organ, cellular or molecular level. This is done by negative and positive feedback mechanism and amount of fluid. This is a regulatory water balance for the digestive kidney are highly efficient conservation of the of water conservation conservators of body water, although kidney filter 180 liter of blood per day. The urine output is in the range of the 1 to 2 liter for 24 hours. Hormonal control of fluid. When water intake is insufficient, water loss is excessive, kidney compensate by conserving water and excreting more concentrated urine. The renal tubules increase water reabsorption in the response to the normal action of ADH, anti-diuretic hormone, released by the pituitary gland under the stimulus of the hypothalamus, and this helps to restore the blood volume. Cells in the kidney respond low blood pressure by releasing enzyme called the renin, which in turn causes kidney to reabsorb sodium through series of events. It is called antigiotensinogen. In the blood, it active form antigiotensine, being a powerful vasoconstrictor. Angiotensin narrows the blood vessel, thereby raising the blood pressure. Angiotensin also mediates the release of the hormone aldosterone, which causes kidney to retain more sodium and thus water. In summary, so they figure a mechanism of restoring homeostasis. Kidney respond to reduce blood flows by releasing the enzyme renin. Renin initiate activation of the protein blood vessel constant raising pressure, adrenal gland secrete aldosterone, kidney retains sodium water, thus increasing the blood volume. Okay. Thirst and salinity influence water intake, apparently response to changes sensed by the mouth, hypothalamus, and nerves. When the blood is too concentrated, the mouth becomes dry and the person responds by drinking. When the hypothalamus detects that blood is too concentrated, it also initiates the drinking behavior. Besides stimulating the thirst sensation, the hypothalamus stimulates the pituitary gland to release the DH. This hormone increases the reabsorption of the water. So, in summary, the amount of fluid in the body is tightly controlled because imbalance can be life-threatening. In a normal individual, the maintenance of water balance is achieved by adjusting both water intake and excretion as needed. There are a number of ways of water input and output. The regulation of water balance is affected by digestive system, kidney, hormonal, and control of thirst. So, in this summary, in this chapter, we learn that what is metabolism. Metabolism is the sum of the all enzyme catalyzed by chemical reaction taking place in the organism. It is also called intermediary metabolism, as substrate goes through the multiple intermediary steps to form the end product. In the all the metabolic reaction participate either in the catabolism, anabolism, or both. Catabolism refers to reaction involve the breakdown complex biomolecules such as carbohydrate, lipids, proteins into simple molecules such as carbon dioxide, ammonia, and water. Anabolism involves step-by-step biosynthesis, simple and complex biomolecules starting from simple precursor. Bioenergetic, we learn what is bioenergetic is the generation of energy by breakdown of the nutrients. Its storage and utilization performing various functions of the cell. Process of biogenetics attempts to unravel the complex network of biochemical pathway involving large number of interwoven biochemical reactions. Living organisms differ markedly in their appearance and they have striking similarly in that they are used in the wonder molecule ATP as the carrier of energy. Free energy available from the hydrolysis of this is used to drive a number of reactions which require an input of free energy. This is done by suitability coupling of the appropriate reaction. The ATP synthesis about 50 to 60 kilojoule per mole. Cell converts glucose to carbon dioxide not in a single high energy releasing reaction but rather in a series of controlled reaction. This type of cellular oxidation requires specialized electron carrier. So this chapter is a over unit 4. So okay, so there are more biology things are here. So you have to understand by, but all the figures are not here in this book, and you have to follow some other books in the other zoology books. Actually, you have to find out these things. Mm, here the so in this unit, in the last unit, you learn the metabolism and differentiate between the catabolism and anabolism, uh, ATP, bioenergetics, and significance, formation, utilization of ATP in the living systems, and homeostasis, and explain the feedback mechanism, and maintenance of water balance in the body. How we can balance water in our body. So overall metabolic process. Okay. So this is all over your 4 units. So In the bioenergetics, there are two conventions which are very important in bioenergetics. The first one concerns the standard state and hence the standard free energy change. Other one is the activity of water molecule in aqueous solution is taken to be unity, although their concentration is about 55 molar. Okay, so this covers unit 1, 2, 3, 4. Unit 1 cell organelles we study, and unit 2 we study the carbohydrate and lipids. Unit 3 proteins and nucleic acids, and unit 4 intermediary metabolism. Okay, carbohydrate lipids, you understood the carbohydrate parts, right? The glycosidic linkage, all the figures you have you seen?
Yes, ma'am.
Okay, so you understand that glyco how the carbohydrates are joined together. You can understand that hydro and glycosidic linkages, polysaccharides, oligosaccharides, starch. Polysaccharides exist in the covalent linkage with lipids, proteins to form complex. They are known as glycoconjugates. And lipids group of naturally occurring organic substance usually formed by combination of fatty acids with glycerols soluble in the organic solvent, ether, chloroform, and highly hydrophobic in nature. Hydrophobic means not soluble in water, insoluble in water. Lipids can uh, lipids are classified based on their structure, complex structures. And fatty acids. What is fatty acids? Have single carboxyl group, long non-polar hydrocarbon chain. Esters of the fatty acids. It contains ester groups. Esters of fatty acids called acylglycerols, tri-substituted derivatives, being neutral in nature and also called triglycerides. The complex lipids contain some additional groups such as phosphate groups, proteins, carbohydrate moiety. These are accordingly named phospholipids, lipoproteins, glycolipids, steroids, bile acids, steroid hormone belong to the class of derived lipids. Lipids arrange themselves into monolayers, micelles, liposomes, bilayers when exposed to aqueous environment. Biological role of lipid include prevention of loss of heat, packing, support materials, etc., preventing coronary heart diseases. Okay, so all the units are over. So we should close now.
Okay, ma'am.
Okay. Okay,
Thank you.
thank you.
Thank you,
Thank
ma'am.
you. Thank you.